Good morning, everybody, and thank you for joining us today for the second day of the Aqua Tech Week webinar series. I'm Alex Shuretsky, and I'm the digital coordinator with Annex Business Media, and I'll be your moderator this week for your event. Before I introduce our speakers, I just want to take care of a few housekeeping items. First and foremost, I want to thank our sponsors for making this week possible. Thank you to BioRouter, Grundfos, Standard BioTools, Hudeck, Balmoral Tanks, Asahi in America, and Cascades. As always, this webinar is being recorded, and a link to the recording will be sent to all registrants 24 hours after the conclusion of this webinar. Questions for our speakers are welcome and can be submitted through the Q&A function at the bottom of your screen. Time has been reserved throughout each presentation to answer your questions. With all that out of the way, I would like to introduce our first speaker for the day, Zach Farrow. Zach is the Senior Field Application Specialist at Standard BioTools. And with that, I'll pass it over to him for his presentation. Zach, the floor is yours.
All right, thank you, Alex. As Alex mentioned, I'm Zach Farrow. I'm a senior field application scientist with Standard BioTools, and today I'm going to talk to you about molecular testing in aquaculture. Hopefully, by the end of the talk, you'll be able to see how Standard BioTools products and applications can help you solve common and complex issues with the X9 high-throughput genomics system. Before I get into any of the meat of the talk or get into why you're actually interested, I want to introduce Standard BioTools just itself. So Standard BioTools, the logo shown here, we've been around for about 20 years. You may not have heard the name Standard BioTools. We were formerly Fluidigm. So if that name rings more of a bell, we are the same company. Formerly Fluidigm, currently Standard BioTools. We're headquartered in South San Francisco and have research and manufacturing in the USA, Canada, and Singapore. But more important than that, we have a global network of field application specialists, field service engineers, product support engineers. So no matter where you are on the globe, you have support if you need it. All right. So most of the talk today is going to be devoted to showing you how molecular techniques work in aquaculture. But before I get into any of the publications or any of the stories, I want to talk to you about the actual technology. So the vast majority of these applications use the precursor to our current technology called the biomark. We currently have an instrument called the X9, and the X9 is our high throughput genomics system. You can run you can run endpoint and real-time PCR on the X9 system. The workflow is 4 pretty much easy, straightforward steps. The first step is to collect and prepare your samples, and that'll be extracting DNA or RNA and then preparing that for downstream use. You'll then also prepare your detection assays for either gene expression, so for RNA targets, or for SNP genotyping, so looking at differences in the DNA. Once you've prepared your samples and assays, you'll transfer everything over to an IFC, or integrated fluidic circuit, or what we call the chip. And the chip is where all of the chemistry is going to take place and all of the reactions are going to take place. Once everything is transferred onto the IFC or the chip, you're going to take that chip and place it into the X9 instrument for loading, cycling, and imaging. So basically, it's an automated step that does all the work for you. After you've collected data, you'll be able to analyze the data with Standard BioTools analysis software, hopefully answering your questions that you initially intended to ask. So to keep in mind, the publications that I'll talk about mostly use the precursor to the X9 called the biomark. So if I call one the biomark or the X9, know that both instruments have the same functionality. They're just used in different ways, and one was used before, the biomark was used before the X9. All right, so now that that's out of the way, I want to talk to you about how the X9 system or the biomark is able to work with aquaculture. Excuse me. So looking at the publications and some of our current customers in the aquaculture space, I've broken down how they use our instruments and our applications in 3 main categories. Those categories are making sure that using our tools to help make sure that fish and other organisms in aquaculture are healthy, making sure that the fish and other organisms are meaty and tasty. That's a little tongue in cheek, but also to make sure that when you want fish, that you actually have fish. And I've broken these down into 2 specific examples per category. The first two I'll talk about is in the health category, and I'll talk about how our customers have used our products, standard BioTools products, for pathogen detection in aquaculture and animal welfare or fish welfare in aquaculture. This first publication is from the Fisheries and Oceans Canada, and they looked at monitoring microbes in salmon. So what they did is they designed a panel, a gene expression panel, to look at viruses, bacteria, and parasites that are commonly associated with disease in salmon. They collect samples from potential sites of infection on the salmon, and then they process those samples. The samples, after processing, are transferred over to the IFC or to the chip, as well as the assays. Everything is automated, and all of the reactions are mixed and automated with in this case, the biomark, but can be used on the X9 system. And once the data is collected, you can tell which viruses, bacteria, and parasites are in the overall population. So once you've identified the common causes of disease, you can mitigate the effect of those diseases on the larger population with whatever means you see fit. And this can be done in a larger or smaller aquaculture setting to make sure that your fish are pathogen-free. The next publication I'll discuss, or the next example, is animal welfare or overall fish welfare. So in this study, this actually happened in a couple of different studies, a couple of different farms. So the authors of this study, or multiple studies, reared fish in different environments. They wanted to observe how water exchange rate in tanks, stocking density, water temperature, and even outside noise levels would affect fish growth. They also wanted to see if they could detect this change molecularly as opposed to waiting till the end of harvest season. So they designed a gene expression panel that analyzed the stress response, the immune response, and metabolism. So they wanted to make sure that their fish weren't stressed, weren't infected with anything, and had a good metabolism. So they were growing at a good rate. So they would collect samples from these fish in different environments, process them, transfer them over to the IFC, transfer those gene expression targets to the IFC as well, allow our instruments to process that and collect the data. Once they have the data, they would compare the gene expression data to physical traits like length, weight, and fight scars, so they could tell optimal environment and stocking parameters. Once they've compared that, they can alter the stocking parameters and then monitor the fish health using molecular testing to ensure optimal growth and health. The panel can be used on juvenile fish as a predictor for full growth. So the panel fits in here by allowing you to see optimal growth before you reach the endpoint. So you can see how big your fish are going to grow before they actually get to that point. All right. So I first started talking to you about how the X9 system and molecular testing can help maintain a healthy fish population through pathogen detection and overall animal welfare. I now want to talk to you about how to make sure the fish or your aquaculture organisms stay meaty and tasty through diet optimization and selective breeding techniques. In this first example, this first publication, in a similar story, fish were fed different diets that had different compositions. And the authors wanted to know how that would affect optimal fish growth. So they designed a gene expression panel that analyzes appetite, growth, and metabolism with the idea that an increase in genes associated with appetite, growth, and metabolism would mean more growth and bigger fish. So they collected samples from juvenile fish and from adult fish, prepared those, and put them on the IFC. They then compared the data to the gene expression data to physical traits such as length and weight, and they could use the panel on juvenile fish to predict the physical outcome. So they could use the gene expression panel on fish that are initially fed the diet to then predict how big they're going to grow. So they don't have to wait till the harvest to see how big that fish is going to grow, to make sure that the diet was actually optimal. All right, now we'll move on to selective breeding. So here is a switch from a gene expression or RNA to look at DNA. So now we're looking at differences in small and specific portions of the DNA. And the customers here developed a 95 SNP panel. And with this panel, they were able to look at parentage so that they could reduce inbreeding in their breeding stocks. They also included SNPs associated with stress, growth, and disease resistance to make sure that the traits that they wanted were included in their breeding stocks. So they would collect samples from juvenile fish, collect samples from juvenile fish, collect data with the panel and see if the traits that they wanted are in those juvenile fish, mate those fish with the desired traits, and they can avoid inbreeding. They would then collect samples from offspring and then confirm that the desired traits are passed on. And this is something that can be applied every breeding season to ensure that your breeding stock has the genes and the associated traits that you want, and make sure that you can reduce inbreeding. All right. So I've talked to you about how standard BioTools products have been used to help maintain a healthy fish population, help maintain meaty and tasty fish, and help maintain that you have fish. Now I want to talk to you about how you can use them to have fish. The first example I want to talk to you about is breed identification, and then I'll move on to a story about conservation. This first one, breed identification. So the paper published here is about rainbow trout. So I apologize, I got stuck there. This is a paper on rainbow trout, and rainbow trout have commercial breeding stocks, a large number of commercial breeding stocks. There are also wild and farmed rainbow trout, and they're difficult to distinguish. So the authors who created the last SNP panel actually decided to use that panel in a different way. So they were also able to use that panel to molecularly distinguish fish breeds. They would prepare the samples from individual fish and collect data using the SNP panel to identify the fish breeds. The data generated provided a greater than 97% accuracy of fish breed assignment. So this gave confidence to the breeders that the fish that they had were the fish that they said they had, and it gives confidence to the consumers that the fish that they're purchasing and consuming are the fish that they want to consume. The final story I'll relate is about conservation, and this is a story from the Alaska Department of Fish and Game. And every year, a number of salmon return to the rivers they were born in, and to get to those areas in Alaska, about 38 million sockeye salmon pass through the Bristol Bay area. And as you can imagine, this is a busy time of year for everyone, humans and fish included. Of those 38 million sockeye salmon, there's not just one type, there are 9 distinct genotypes. And of these 9 distinct genotypes, they don't always come in the same ratios. So to ensure that fish and the same number of fish keep coming back year after year, it's important to make sure that no one stock is overfished. So in an effort to conserve fish and to make sure that fish return in the same numbers year over year, the folks at the Alaska Department of Fish and Game will genotype these 9 different stocks to make sure that no one stock is overfished. And they have about 1 week to genotype all of these fish and then provide that information back. So previously to using our technology, they would need to genotype a whole ton of fish in one fishing season, which related to about 384 well plates. I don't have that number off the top of my head, but if you multiply 1,300 by 384, that's a very, very large number. So they would have to process 1,300 384 well plates in about a week. Using our technology, they would reduce that number down from 1,300 to 54. So just looking at the numbers without having to know anything about either running chips or running 384 well plates, 54 seems much more manageable than 1,300. So not only is it a more manageable number, it's a reduction in time, it's a reduction in effort, and it's a reduction in plastic that's also going back into the environment. So there's a whole conservation effort, not just for making sure that fish return, but that they have an environment to return to. And with that, I'll move on. And hopefully throughout the talk, I've been able to show you that standard BioTools products and applications can help keep fish healthy through pathogen detection and overall animal welfare. making sure fish are meaty and tasty through diet optimization and selective breeding, as well as making sure when you want fish, you have fish through breed identification and confirmation, as well as conservation efforts. So now, hopefully, whenever you think of aquaculture, you now think that there's a molecular method that can help you with it, and the X9 high throughput genomic system can help you with those methods. So if you're interested, please let me know. Please contact me, Zachary Farrow at standardbio.com. I can also direct you to the sales and ordering. And if you don't remember my name or the sales and ordering name, John Kupcho, you can reach out to tech support and they'll get you the information you need. And with that, I'll thank you and end my talk.
Awesome. Thank you so much, Zach. Maybe if you want, you could put the contact information slide and just leave that up while we answer some questions here.
Absolutely.
For sure. Okay, awesome. So I got a couple questions here for you. If the X9 system is also able to generate libraries, is the X9 system, also able to generate libraries for sequencing?
Yes. So the X9 system can do a couple of different things. So it can do real-time and endpoint PCR. And a new feature that recently came out on the X9 is next generation sequencing library prep. So it doesn't do the sequencing for you, but it will process the libraries.
Awesome. Okay, thank you. So in your presentation, you mentioned samples were tested in these studies. What type of samples were they?
So the samples that are going to be used here are going to be extracted DNA or extracted RNA that's been reverse transcribed. The samples will come from thin clips, is going to be the most common sample type from fish, where you just take a small part of the fin and then you extract the DNA. If you're doing something like pathogen detection or gene expression, you need to take the sample from whatever site is going to be infected potentially. So that will be for pathogen detection, or for gene expression, you'll need to take it from the organ or that you're interested in, or blood.
Okay, perfect. Thank you. Are the SNPs and assays introduced to the chips separately, or are they introduced together?
I misspoke. I believe those are the same. If I'm getting the question correctly, I think those are the same thing. So the SNP is a sorry, I'll try to do my best to answer that question.
No.
We have the IFC itself. You place the samples on one side and the assays on the other. So one side is going to be individual samples. So it's going to be an individual DNA sample from an individual fish. The other side is going to have individual SNP assays. So it's going to be detect once each well. So you have up to 96 detectors, and you're going to have an individual detector in every well, and each detector is going to look at one SNP. So each sample will get mixed pairwise with each of those SNP detectors. So you have a large panel running at once. Did I answer the question?
I think so.
Okay.
We can move on to another one for now.
Okay.
If someone wants to follow up, they can.
Okay.
You mentioned multiple panels for testing. How can I run the same panels for my studies?
Sure.
And can I customize them? The two-part.
Sure. So all of the examples that I discussed here are from published research or published papers or protocols. So all of the targets and panels are publicly available. We don't offer them, but we can help customize the gene targets and the panels that are listed in these publications. We can help make sure that you get what you need and customized for your specific projects.
Okay, perfect. Thank you. You may have already answered this one, but I'll still ask. I heard that Standard BioTools launched a NGS library prep feature. Can you confirm that?
Yes, it's on the X9. You can absolutely do that.
Okay, perfect. Another one here. How much does the equipment cost, and how much will it cost for each sample?
That is a good question, and I am going to have to refer you over to sales. So if you could, whoever asked that, if you could follow up with either John Kupcho, shown here under sales and ordering, or reach out to tech support, they'll get you to the, either one will get you to a salesperson and they can answer your question.
Perfect. Thank you. Is the technology quite developed to adapt to other species of fish or shrimp?
Oh, absolutely. You can use, so as long as you can get DNA or RNA, you can use the system. I had, so shown the publications that I used showed a lot of salmon, but that's just because people like salmon as a common fish to consume. But we also showed rainbow trout. I've read papers where people have looked at shrimp, looked at oysters. I think I've seen octopi. So really, honestly, if you can get DNA, you can run it on here.
Perfect. Okay, awesome. Does standard BioTools sell assays?
Yes. We do have an assay design service, so we can help you design those assays for your specific organism and your specific workflow. We also, if you already have assays that you're using, we can help port them over to our technology.
Okay.
So yeah.
Thank you very much, Zach, for taking the time out of your day to present this for us. We really appreciate it.
Thank you for the opportunity, Alex.